Hey guys, welcome back. Third and final video for Chapter 4 cells. Let's jump right into it. We're starting off with the cytoskeleton. Last video we covered a lot of ground, for sure. We went over a lot of organelles, and this time we're just going to kind of wrap it up with a few other points for things that are holding cells together, and we're going to call it good. So, the cytoskeleton, if you break down the word, cyto means cell, and of course you guys know what a skeleton is. You have all the bones holding up our body. So a cytoskeleton is the cellular skeleton, the support structure for a cell. And so this is an interconnected system of a lot of protein filaments. In biology, if you don't know, guess protein, because that's probably the right answer. Proteins do almost everything, and one of the things they do is provide the structural support for a cell. Some of these proteins are permanent. They're always going to be in these locations, but a lot of them are temporary. They can polymerize on their own and then depolymerize on their own. So it's sort of an odd thing. They can build themselves up from little tiny structures, and then they can take themselves apart. So it's like the 2x4s in your house. They hold your house up. But if you were a cell, those 2x4s could fall apart into little splinters of wood, move themselves to a different location, and then reassemble from the little splinters of wood back into a 2x4. So a cell is a weird place to live. But they're going to reinforce parts of the cell, organize things in the cell, move cellular structures, and sometimes they can actually move an entire cell, which is amazing, and we'll talk about that in a little bit. So there's 3 types you need to know: the biggest, the smallest, and the middle ground. So the biggest, which is, the widest, are microtubules. Microtubules are hollow cylinders made of proteins called tubulin, and they form dynamic scaffolding for cellular processes. Dynamic means it's changing all the time. Scaffolding is a support structure you can stand on if you're doing siding on a house. And so these guys will assemble themselves somewhere where the cell needs to move something. And then when the job is done, they'll disassemble themselves and go somewhere else. Microfilaments are pretty small, thin little guys. And remember this because filament is the thin wire that glows in a light bulb. So when you think small, think filament. Smaller than a tube, because they both have the word micro in them. And so microfilaments contain the protein actin. If you've taken anatomy and physiology, it's going to sound really familiar because he's like half of the job of moving all the muscles in your body. And these little proteins that are called actin are going to make up the microfilaments, and they're going to do a lot of work in the middle of the cell. And then we have the intermediate filaments, which are between they're bigger than a microfilament, but they're smaller than a microtubule, and they're just going to do a lot of work maintaining the cell's shape and then the structure of certain tissues. And so here we have it. Microtubule, he's this big circle, rather cylinder. The microfilament, these little tiny actin dimers, coming together. And then we have our intermediate filament, which is this coiled sheet of proteins. And so here we have, this is such a cool picture. I had told you before that they, would add color into electron microscopy to make it more apparent what's going on, and this is what they've done here. So a fluorescence micrograph showing these microtubules that are yellow and microfilaments that are blue in the growing end of a nerve cell. Kind of a cool, very cool picture. And so these guys are guiding and supporting a nerve cell as he grows out. And so that's one of the many jobs they do. But you can see that a lot of that cell is made of these supportive structures. So kind of cool. So one other thing that we have with our structures in the cells, our cytoskeleton, are these little guys called motor proteins. Remind me in class, and I'll show you the Harvard cell animation. He'll be a good review of this stuff. And they have a motor protein on there, and he's just so cool. My other classes have mostly called him the little walker guy because he just walks. That's like what he does. But this thing is just a protein. He's not alive. He has no brain. He is just a protein. And what he will do is he will take a vesicle or a little vacuole or an organelle or something like that, and he'll grab on with one end, and then he'll stick his feet onto this long microtubule, and he'll move. he'll form chemical bonds right here, and he'll lift his other foot up and put it forward and bond there, and we'll weaken the bonds in the back, and he'll just alternate feet, and he will walk up this microtubule, and it's pretty amazing. He's got to burn some ATP to do this, but he can transport things around in the cell, so kind of a cool job. So, cilia and flagella are structures we've talked about before. Flagella being the long whip-like tail, and cilia being these short finger-like projections out of bacteria and cells that can kind of beat and either move a cell or move structures that are outside of that cell. And these guys are made up of little supporting structures very similar to our cytoskeleton we just talked about. And so these guys are formed from these microtubules that we just talked about in these 9+2 arrays, which means there's 9 on the outside and 2 on the inside. And the cilia are generally short little structures that kind of do the wave, and they can move fluid around, and they can move all sorts of things, eggs down fallopian tubes, mucus up throats, that kind of stuff. And then flagella are long whip-like tails that you might find on sperm cells, for example, or bacteria, and those long whip-like tails are going to rotate, and they're going to move the cell around. The microtubules, they grow from kind of this barrel-shaped centriole that holds them in place, and he'll remain in the cytoplasm below as sort of like a basal body or a basement membrane to build on. And they'll even do that when they're pulling DNA apart for something like mitosis. They do that as well. So those centrioles are rather important, and we're just kind of coming to understand now all the different jobs that they have and how important they really are. If you look in your textbook, which really isn't that old, it's so funny. The Gen Bio textbook we have here at school, it says something like the centrioles at this point in time, we're not certain of their function or something like that, and the book's not that terribly old, so it's sort of funny. We're becoming certain of their function, let's say that. So we made mention before that these cytoskeleton components could be used to move structures or sometimes used to move the entire cell, and that's what we're going to talk about now. Pseudo is false or fake. You might have heard maybe in like an English class, some authors will write under a pseudonym, a false name, and so pseudo's false, and then pods, like a tripod for a camera has 3 feet. So a pseudopod is a false feet or false foot, and these false feet we see in amoebas and other big cells that need to move around. And so a few of our eukaryotic cells will make lobes in their cell membrane, and they'll bulge outward to the direction the cell wants to go, and then the rest of the cell can drag its contents in that direction, and they can move the cell. We can also form pseudopod around smaller cells and use those to eat smaller cells. That's like an amoeba that might eat a plasmodium or something like that. And then we have elongating microfilaments. So we're aware of microfilaments at this point. Force that lobe to just push out in a steady direction. And then these little motor proteins that are attached to microfilaments are going to kind of follow behind and drag that plasma membrane with them. And so we can really get this kind of creeping amoeboid motion of amoebas, and just a few other protists can do this as well. So it's kind of a cool example of moving a whole cell. So let's look at our cell surface. A lot of cells will secrete materials outside of their plasma membrane. It's not like there's a cell and then outside of the cell there's just water and that's it. It's not like that, really. The cells secrete proteins, and they secrete lipids, and they secrete sugars, and they build what's called an ECM, or extracellular matrix. And the extracellular matrix is not living, but it's a bunch of proteins and sugars surrounding that cell. And it depends on the type of tissue, but the extracellular matrix can actually make up a huge amount of space in a certain tissue. Like bone is mostly extracellular matrix. You know, broken bones, they're not cells mostly. They're extracellular matrix that's breaking, and the cells will grow new matrix to heal it. And so extracellular matrix is a super cool idea of cell walls, cuticles, and even just sheets of proteins on the outside of cells that really give us a little bit of an environment to keep cells stable, and also a buffer between them and the rest of the world where they can adapt to stresses just a little bit better with that extracellular matrix. And if you've got some time on your hands, I'll point you guys towards a video. It's this old like Discovery Channel documentary where they use extracellular matrix to help organisms and people grow back lost structures, grow back lost limbs, and it's really neat. They get some guy, you know, they grow back like the end of his finger or something like that. And it's just cool. They just use extracellular matrix to do it, so it's kind of a fun idea to be familiar with. So let's move to cell walls. When we're talking about cell walls, animal cells, you and I, you know, we don't have cell walls, but plants do, and some fungi do, and a few protists do as well. So when these guys are building cell walls, they have a primary cell wall and they have a secondary cell wall. A thin, pliable wall is the primary cell wall, and they're just pretty much secreting cellulose around the plant cells that are brand new. And then as those cells age and get stronger, they'll start to put lignin on that cellulose, and that lignin will make it really tough, a little more watertight, a little more bug resistant, and you get a really strong plant cell wall. So what we think of mostly with plant cells is cellulose, and that's fine, but know that there are lignin and there are other waxes and some watertight cuticle stuff that's going to be pushed out into those cell walls. So cuticle is a type of extracellular matrix that cells secrete at the surface of their body. Plants do this with waxes and a few proteins, and it will help them retain water, and it will also make it really hard for insects to chew through that leaf. And then there are a few animals that create cuticles as well. Crabs, spiders, animals that you step on that make a crunch when you crush them, mostly use what's called chitin, and it's actually a carbohydrate. It's not even a protein. I tell you to guess protein all the time, but this one's a carb. Isn't that weird? A polysaccharide, a big sugar that you can use to make a cockroach shell. How nasty is that? But there you have it. So cuticles, they're not just for plants. Now, cell junctions are a little tricky. They're important to understand for how cells communicate. They're just not the most exciting thing in the world. Cell junctions are where one cell meets another cell, and they allow cells to interact with each other in the environment. Super important. Okay, not super fun. Super important for our survival. Absolutely essential. But in plants, these guys have little plasmodesmata, and they're like little channels that connect one cell to his neighbor. And this is cool because for plants, they can push sugars and fluids into pretty much all the cells without too much trouble. And when a plant gets dehydrated, it's not like one cell wilts and dies. You know, the whole plant starts to wilt together because every cell is connected to his neighbor. And that's an important feature for plant cells. We call those plasmodesmata. Because botanists like to feel smart and they give them really long, wordy names. Animals, on the other hand, do not have plasmodesmata, but we have something similar. We have 3 types of cell junctions. We have tight junctions, adhering junctions, and gap junctions, but we don't have plasmodesmata. So, if you're an animal, or fish, or dog, or human, or whatever, you got a few. Tight junction is fastening together the plasma membranes of adjacent cells, and what this can do is basically give you a watertight barrier. So the whole side of the plasma membrane from both neighbors are just kind of stuck together. Adhering junctions is like skin cells being stuck to the basement membrane that has the blood supply. It's really just one side of the cell that's adhering, and the rest of the cell is more or less free. Then we have the really cool one. These are gap junctions. This is kind of the human equivalent to plasmodesmata, and these are closable channels that are going to connect the cytoplasms of our adjoining animal cells. Gap junctions are the reason that heart tissue, heart muscle can respond from one single source of a stimulus, the whole area of the heart that it's targeting, like left ventricle, can all contract at the same time because all of those cells have open lines of communications with their neighbors. So gap junctions, all of these are important, of course. I mean, I enjoy the fact that my skin's more or less watertight, don't get me wrong, but gap junctions are just incredibly important and pretty amazing structures. So gap junctions, keeps your heart beating. We like that. Here's a picture. It's nothing too fancy. Tight junctions, you basically sew these together. Gap junctions, we have a channel. Can be open or closed for ions to flow through. Adhering junctions, we take one end of the cell, we stick it to a basement membrane on one end, and the rest of the cells, just kind of up there with its neighbors. And then here's your plasmodesmata in the plant cells. Just kind of cruising through here. It's a free area for waters and sugars to kind of flow through from one cell to another. That's about it. All right, so this chapter ends with kind of a weird little note on the nature of life, which I feel like we kind of addressed in chapter 1, but okay. We define life by describing sets of properties that's unique to living things. In your book, it says life is squishy. That's their simple definition for it, so I guess that's good. Life is a property that emerges from cellular components, but a collection of those components in the right amounts and proportions is not necessarily alive. And so that, I think, is cool for you guys to kind of understand because, as young Christians, and we're going to talk so much about evolution in this class, and that's fine. That doesn't bother me, but it is important to understand that life is more than the sum of its parts. If you took a beaker and you put amino acids in there and some carbs and little proteins and some lipids and you mix it all around, you'd have the components, but you wouldn't have life, if that makes sense. Life, we define it by saying it's made up of this and this and this and this component. But if we just had this and this and this and this component, nothing would be alive. So biology's it's a cool thing to study. It really is. It's a little more than just 2 and 2 is 4 when it comes to living things. There's a little more to them. So life, for it to continue, it will continue only as long as we have a continuous flow of energy to sustain its organization and to sustain its growth. So life does depend on energy, but individual components plus energy doesn't equal life, right? We call those emergent properties. And as simple as it is to, I guess, kind of discuss it, it's a little bit harder to conceptually really wrap your mind about just how incredible, not to sound like I have a huge ego, but just how incredible living things are, how incredible you and I are, the fact that we're alive. It's pretty amazing. So living things, what do? They make and they use organic molecules of life. Yes, we do, right? The plants make the sugars, the humans make the donuts, and we digest the donuts to make energy for us. So there's our living things. They consist of one or more cells. We got that with cell theory. They engage in self-sustaining biological processes such as metabolism and homeostasis. So this basically means regulation is what we're talking about here. Metabolism's breaking down food, building up useful things. Homeostasis, you shiver when you're cold, you sweat when you're hot, and your endocrine system takes care of the rest. You change over your lifetime. For example, you grow, you mature, you age, that kind of stuff. Living things need to use DNA as hereditary material, and they need to have the collective capacity to adapt to the environment over successive generations. So we throw some evolution in there at the end, just icing on the cake. You got to adapt to your environment, right, to survive. If you can't survive and produce offspring, you lose the game of life. And if you survive and produce offspring, you win the game of life. And that's really the basis for your big idea of evolution and change. So we'll talk more about that. So, points to ponder. Here we go. End of the chapter. Everyone's excited. Why do bacteria have ribosomes when they lack other organelles? I think you could probably answer that. Actually, you should answer that and we'll talk about it in class. Please write this down. Write your answer. Why do bacteria have ribosomes when they lack other organelles? Question 2. Does the cytoplasm have functions of its own, or is it just a filler in which other organelles float? And then finally, what organelle could be compared to the control center of the assembly line in a factory, and why? So, a couple questions, not so bad. 1 or 2, a little trickier. So take some time, answer those the best of your ability. If you want to Google it, add some information, absolutely do. And I thank you guys for that.